Okay, so I would like to introduce our next speaker. Our next speaker is Joe Trask. Joe is a product line leader and senior feed application scientist in the cellular imaging and analysis group at PerkinElmer. Joe will be giving us a talk on high content screening assay development. Welcome, Joe, and the stage is yours.
So yeah, thank you, Serene, and thank you for the opportunity to present here today. Good morning, everyone on the West Coast, and good afternoon to the East Coast folks, and good evening to any European colleagues that may be joining us today. So I wanted to start just kind of breaking up the talks a little bit and just ask you some trivia questions about the assay guidance manual, just to see if anybody actually is aware of some facts about the assay guidance manual. And you can see the first one, I already have the answer up because I was moving my slides, but how many pages are in the assay guidance manual? And the answer is 1,315, which was pretty astonishing, to me that we had that many pages. And so this will be a little, light reading for you at nighttime. And with that, how many words do you think are in the assay guidance manual? Do you think it's about 500,000, over 1 million, or are there really too many words? There are really no words to tell you how valuable the assay guidance manual is. And the answer is, it's close to 500,000 words, and they're in the assay guidance manual. And the other question I have for you, what is the most common word used in the assay guidance manual? So I put 4 words up: cell, screening, compounds, and assay. So just take a minute to think about that. Cell is mentioned 3,343 times. Screening is mentioned 1,723 times. Compounds is mentioned 2,900 times. And assay is mentioned over 7,000 times. So if you guessed assays, which is, the assay guidance manual name itself, you were correct. So, this is kind of a composite of word cloud just to show you that assay and assay are the most common words used in there. And if it's, using the analogy of Finding Waldo, one of the things I want to talk to you about today is high content screening. And you can find HCS is actually quite small in the context of other words used in the assay guidance manual. So I put my contact information here on this slide, so you can contact me at joe.trask@PerkinElmer or at highcontent@gmail, or you can connect through LinkedIn. I'm sure to happy to address any questions or comments that you may have. So today I want to talk to you about some best practices and approaches to consider in high content imaging assays. So the bottlenecks really in high content assays, today really are around, really 3 things. And I really want to mention these 3 things are around image analysis. And I think this is really for people who are just entering the field of high content screening. Maybe they're biologists and they don't really understand how to develop and run image analysis algorithms. I think the newer thing that's in the field today are learning the concepts and workflows for using machine learning, AI, deep learning, and neural nets. And then the last thing is assay development. And this is not in any particular order. There are people that understand assay development well, but may not understand the other concepts of machine learning and AI or image analysis. So it really depends on where you're coming from. So I'm going to try to tackle some of these and give you some resources on how to have some best practices in these particular areas. Now we have in 2012, so it's been 8 years ago, we first published the assay development guidelines for image-based high content screening chapter. And so please use this. You can go there and get some detailed information about how to develop an assay, how to validate that assay for high content screening. There's also an advanced assay development and guidelines for image-based high content screening and analysis chapter that was submitted shortly after that by Ann Carpenter and Mark Bray from the Broad Institute, that goes a little bit more about using tools in Cell Profiler for doing your high content imaging assays. There's a comprehensive chapter on NF-kappa B translocation. This goes into a lot of details behind the scenes things, some of the things that Terry was talking about on the cell passage number, also looking at different lot-to-lot variation that you may see for an assay. So very comprehensive chapter. And then there's a high content screening chapter with primary neurons that was submitted by University of Miami, Vance Lemon and John Bixby, who go into some details about that. There are some new chapters that are under construction. There's one on machine learning for image-based screens. So hopefully that will help people understand these concepts a little bit better. That has been under review and should be coming out relatively soon. There's also an interference and artifacts chapter that we're working on right now and should have it submitted by the end of the year and likely be published in the AGM in the first quarter of next year. And then we're also working underway chapter on 3D imaging. So just want to mention a little progression of high content kind of over the years. And part of this, when we first started in this this discipline of technology, we used to call it computer-assisted automated microscopy. And I took this is a picture I found. This is a picture I had from 1993 when I was at the Ohio State University Comprehensive Cancer Center, where we had a Nikon scope hooked up to a video camera. So very slow and low resolution camera. But it was an automated imaging analysis system. No automated stage, nothing fancy about it. Very low throughput on the system. We had to program things ourselves, so it's very cumbersome that way. But in fact, it had machine learning on it. So machine learning even dates back to this time period. Very small, low processors, and some of the older generation like myself maybe remember some of the old computers from that time period. Today, we call this evolution to high content screening, high content imaging, image cytometry, high content analysis. And this is the most recent system on the market, which happens to be from the company I work for, PerkinElmer, which is the Opera Phenix Plus, which has liquid handling on it. But the point is that these things are very fast today. They have very high resolution, have cloud-based analysis tools on it, machine learning, and very fast processors to interrogate this data. Thinking about those applications, and this is something I want to go over today. Some of those top applications for high content today are really cell painting, 3D cell models. I still think in the field of immuno-oncology are still big players. Virus and vaccines, of course, with COVID, there's a lot of people that have transitioned their high content systems to work on this particular project. So again, and I show this slide almost every time I give a talk for the AGM, and I think it's important that people understand that the images that we're generating on the system is the data. So regardless of those images that you generate, the good, the bad, and the ugly, as I call it, is that it's important to make sure that you capture a good image. Because we're measuring things such as the size, the shape, the intensity of these proteins that you can actually see in these particular images. We can also measure the texture, the dynamics, the kinetics, some of the live cell imaging that Terry was mentioning. We can measure that by high content. And definitely the distribution or the translocation maybe events like NF-kappa B, as I mentioned earlier. There's an example of a primary neuron, for example, or you can look at differentiation that's occurring in these cells. Rarely, as you're going to see, there's a live brain slice image there. Rarely you're going to see homogeneous type cells that are going to occur. This is an example of mitotic index or phosphohistone H3. And this one actually has some fluorescent artifacts in the bottom right panel, but it looks very similar to maybe some of the other treatments. And we're going to have a chapter to talk a little bit more about that. Unfortunately, as good as we are as HCS practitioners in the field and the art of doing this work, we're gonna find images like this, and we're gonna generate images like this. Now, this could be due to a bad plate. It could be cells that were contaminated. It could be the plate was improperly positioned inside the stage, or some optical or some other optical reason. And so if you do find an image like that, the important thing is that you need to find ways to remove that from your data set. So it's important to reproduce data, and I think that's the theme of this entire AGM workshop. And for high content workflow, we start with a cell model. So we want to make sure we have, good cells, as Terry mentioned in the previous talk. The vessel selection or microplate selection is going to be very important. I'll put more stress on that in a minute. We use automation, but not always, but definitely for screening. We would like to use it for reproducibility. And we use this for seeding your cells, compounds, reagents. We're going to use some image analysis system to capture that. We'll undergo some image analysis process, and then we'll go through the data interpretation. I think what's changed in the last 5 years or so is really the addition of using machine learning and AI at all phases of this, but especially in the phases of image analysis and then also for the data analysis interpretation stage. But it's also being used to make better decisions about which compounds I use and what cell lines I may want to use for the process. And very important to make sure that we generate good images, because the training sets that are being used for the machine learning and AI algorithms are really dependent on how good those images are. So as I mentioned, the microplates, I'm going to stress this, and there's a recent chapter that the AGM published this year that you may have heard about, but it's microplate selection and recommended practices in high throughput screening and quantitative biology, and there is a section on high content in there. The important thing is that we want to make sure that we select the best plates. It's very critical for high content imaging assays that you might imagine. So if you are going to be using a new microplate that's maybe hasn't been out there, or even if you're looking at new lots of microplates, you may just want to inspect them to make sure that all of the fields that you're going to be viewing in those plates are accurately being, the resolution is good, the image looks, resolution looks good, and the algorithms are measuring what you're expecting. This is an example of a new plate that we're evaluating. This is when I was at the Hamner Institute. This, you can clearly see here, there's some cells that are out of focus in this particular area, and mainly, and also really on this entire side. I'm going to turn the laser pointer on here. So mainly on this side right here, you can see the out of focus cells that are actually present in this particular well. And why does that matter? So it matters because when we go to the image analysis algorithm, what we're gonna find is that when we have in focus images, we can clearly segment those objects. But when it's out of focus, you can clearly see that there's an issue. And you don't know that because we're generating thousands of images. So we want to make sure that we have the best quality images possible before we undergo the image analysis process. So again, this is part of that new chapter that we've written, this particular graph, and it really is to help people understand about the bottom thicknesses of the microplates they may be using and the effects it may have on the optics they may be using on their instrument. So if you're using thick bottom plates like a 0.7 millimeter, this is what it looks like with the 10x objective. And then when you go to a little bit higher numerical aperture, which is not really conducive for this particular plate, you can see this image is definitely more blurry, and you're not getting the resolution that you would get if you're using a thin bottom plate that is corrected for the objective lens that you may be using. So, it's really important to match the objective lens with a microplate bottom thickness. So there were two of the topics I really wanted to spend today time on are really on 3D high content imaging considerations. There's really 5 things to talk about. I want to talk about a little bit about generating uniform spheroids, cysts, or organoids, obtaining the best quality images, optimizing the image and data acquisition process, and the importance of having 3D visualization when you're doing 3D cell model systems, and really how to get more out of your 3D model system. And then we'll follow up by spending a little time talking about cell painting, which is another topic that a lot of people are quite interested in. So to generate the spheroids, now they come in a lot of different shapes and sizes. They could be spherical in nature, like this one here. It's a nice pretty picture. That's the sphere I'm holding in my hand. Or it could in fact be something that looks like a hockey puck or an oblate formation, or could be prolate kind of formation, more looks like an egg shape. And so these spheroids are going to come in different sizes and shape. Now, there's different methods to definitely generate these 3D cell model systems. This is a nice review that came out a couple of years ago, but it goes into some of the some of the areas that people are using. So you may use 2D cultures, typically going to model layer showing here. But when you go to 3D, we can use many different things. We can use hanging drop, gravity plates. You can use ultra low attachment plates, which is likely the most popular one. I'll show you an example of that. There's also people using magnetic beads to force assemble the cells together to form a spheroid. And then there's also the hydrogels, the matrigels. And so this can be biological hydrogels, or they could be synthetic derived hydrogels. And there's a group of people who are using micro pattern plates also to create these spheroids. And you can also have microfluidic devices, including organ on the chip type models that also can be 3D. So for the ultra low attachment microplates and phenotypes, and this is a pretty simple process, and I think this is why most people will venture into the 3D space. They'll use this particular method. So they're going to take cells from a culture flask. They're going to put them into a 384 or 96 well ultra low attachment plate, and over a couple of days, or 2 or 3 days, it's the standard. I think most people are practicing today. You're going to form a spheroid in there. So cells are they're forced to go into the bottom of that well, and then they start to self-assemble there. And here's an example of 4 different cell lines, and really I wanted to show you these because there's different phenotypes. Not all cells are conducive to form spheroids in these ultra-low attachment plates, so you really have to go through your due diligence to make sure you actually form in the spheroids themselves. And so if you look at something like a prostate cancer cell line, the G145s actually work pretty well. When you go to the HeLa cells that Terry's already mentioned, you can clearly see they form nice spheroids over a couple of days. But when you get in HepG2s as well, but when you get to the MCF7s, a breast cancer cell line, you can see they're more loosely assembled, and this may not be the appropriate cell line of choice for an ultra-low attachment. You may have to move those to a hydrogel or matrigel if you want to use them, or use maybe an alternative cell line such as MCF10s to see if that may work for you. So in the hydrogel or matrigel and certain phenotypes you may be getting there, the cells and the spheres are randomly kind of localized in the media or the substrate itself. So they typically are going to be forming different size spheroids or organoids in there. And so when we start to image these, one of the challenges with these is the position of where they are actually in the Z space, but also the size of them. So we can image it here, and we can capture a series of Z stacks to identify where that particular object is. When we move it over to another field of view, and you can see it's a much smaller object. We can identify that, but it's in a different Z plane, but we're still capturing the same number of Z stacks. And then we can get a field of view where there's actually nothing. This happens quite often when we use these hydrogel, matrigel scaffolds. And then here's one where we just partially clipped, part of that particular, organoid, and then maybe clip the other side of it using this. So those are some of the challenges that we're faced with when we do this, but nonetheless we're capturing entire large Z stacks, and so we have to try to find ways around that. And I'm going to show you some ways that we can approach this challenge. So obtaining the best quality images, and this is really I typically have gone into the past, I've gone into some more detail about the instrumentation that we're using for capturing 3D images. I'm not going to do that this time. But I do want to stress that we can look at using wide field or confocal, and that's typically most of the commercially available high content screening systems are out on the market, and that's what people are using. Now there is LightSheet. And LightSheet is, and people may be familiar, probably more so in academia than they are in the pharmaceutical industry. And that uses more, you can see here, going through the cell on kind of a sideways, kind of a slit scan view. Now with the adaptive optics LightSheet, you can get down to about 20 nanometer resolution. But for the confocal and wide field, we're stuck to the fraction barrier limit of about 200 nanometer resolution. We have to, we're going to work within those boundaries. So the image quality, it does differ though. So wide field, you get a large illumination of light coming through, and so the resolution is not as good. And we go to confocal, where there's a pinhole there, we can get rid of a lot of the out of focus light, and then you can improve that particular resolution. Now we increase the numerical aperture. So the numerical aperture on this particular lens is at 0.6 nanometers. We increase it to 0.75. You can see you get slightly better resolution. And by the time you get up to a numerical aperture above 1, which is with a water lens, you can see a much better better resolution here. And these were done with not live micro tissue. This is actually liver microtissue. So some of the reagents and probes that can be used in 3D models, and I'm not going to go too much detail here. Just to say the important is the accessibility, how well you're staining your cells. You need to make sure that the dyes are getting in there. Beware in the tumoroid type models that you may have a hypoxic core that may be present there, and so you may want to consider probes that may identify where those hypoxic cells are. Many of the probes that people are using in this space they're adopting these from two-dimensional assays that have been typically used, and some of those do work. We know that nucleic acid dyes like ARCS and DRAC5 work pretty well for 3D cell model system. Calcium works pretty well. The MITRE tracker and some of the other organic probes seem to work pretty well. But antibody staining, we're not sure about. The thing we have found out with some of the typical probes that we used in 2D, the concentrations are increasing, all the way up to 10-fold stronger. And then part of that is probably getting it in there. It could also be due to the limited penetration of light to actually excite the probe. Now, the other thing that people have been looking at is tissue clearing agents. This is something that was adopted by the histology community for looking at some of these 3D cell model systems. And this is an example here of a study that was done that compared different clearing agents to see what the difference was between a PBS, just a control, and then clearing over time. And some of these probes require, and this is the scale reagents, and there's some other ones here I'm going to show. Really, it's a combination of maybe some sucrose, urea, Triton, and some other things that are in there. We have found that it takes actually several days to get good clearing that occurs, but some of the other ones, some of the alcohol-based clearing agents actually work quite fast. And but you can see there's some differences in the cell morphology there. And why the clearing can be important for you it comes back to the image analysis. So when we actually segment these objects in 3 dimension, you can see here we're actually doing a cross section because we're not able to penetrate all the way through the 3D sphere. So now you get this, we have this cone-like shape that's shown here. So you can see this is a bit hollow in here. That's not a necrotic core that you might expect, even if it was a tumor cell. This is actually empty space. We wouldn't know that unless we actually used a probe to identify necrotic cells. When we look at something that's been cleared, like scale S4, you can see we can image the entire sphere, so we can identify all of these objects in all spaces. So here you can see the difference is between 633 nuclei up to over 2,071 nuclei here. But you got to be aware of the changes that can happen when you use clearing agents. And this is a paper that came out earlier this year. It's actually a pretty good review on some of the clearing agents that are out there. So you can see PBS actually has the largest sphere. So all of these have some maybe slight effect, but some are statistically significant, such as the clear T2 and Cytovista are actually quite smaller, that's shown here by the change in the diameter of the cell. So just be cautious if you do use these reagents, that they could alter the morphology from which the starting material that you started with. Again, the clearing agents are used on fixed cells. These are not live cells, so the cells are going to be fixed before they undergo the clearing. So in the 3D imaging process, typically we're going to take a 3D cell model system like here. We're going to take a series of Z stacks as shown here, and there's different options that people in the field are taking. First, they will generate a montage, if you will, an array of all these different Z slices at different Z heights so they can actually see it. So here, starting at the bottom, which is right here, so that's going to be the first one. And as you start to penetrate through, maybe you're somewhere here in the middle, in this particular range, and then maybe there's some limit of light penetration into the 3D cell model. So if you did not use clearing, or if the sphere is actually quite large, the probes, and there's a lot of different factors that could be a reason why you can't image through the entire thing, the cells start to get a little bit dimmer, as shown here. So you can take all those images from that array and actually collapse them, and we call that a maximum intensity projection image. And then I'll talk about how we actually do the processing of that image for the analysis and the data. You can also take that same stack and actually do a volume reconstruction of the entire 3D spheroid. And then there's a group that will actually use deconvolution. So deconvolution is an algorithm, really. It does a point spread function to clean up the data. It's very CPU intensive and very sometimes difficult to reproduce. So on the reconstruction maximum intensity projection image, this is an image, this is a 2D image. Now you've taken a 3D stack and made a 2D image out of it. The processing time is just slightly longer than 2D only because you had to collapse those images for the analysis. You do lose that architecture in a three-dimensional space, and so that information is not preserved. Now, when you go to the individual Z slices, that's another way of approach this. So you could take each one of those Z slices that were in that panel that I showed you and do the analysis on that, and then you could do a sum of the mean of all that information together. Again, you're going to lose the context of that information that's rather in the 3D cell model. And then the last one is the volumetric Z images. So this is a process where you look at everything in X, Y, and Z. It's very slow. It's a very process intensive, but you do preserve that spatial information in all directions. So, so when we talk about that process, so I want to show this because this is an example when we start to cut through a sphere and how we can actually identify those objects. So this is an example again of that same panel. So I'm starting here at the beginning, so just so you can see, we can start to segment and look at these individual nuclei in this particular case, and identify those particular objects in a solid sphere. And then we cut through, so we're somewhere in the middle. So now we can identify each of those individual nuclei as we start to move through. This is a tool, and I'll talk a little bit about visualization in a moment, but this is a tool where these little white balls are actually allow you to actually rotate, move it, and penetrate through the particular object. And then you get to the bottom, and then you can actually see there. So that's important to know that you're able to see and segment, that process. So what's going to be most important for you? Do you need to capture all of these Z slices to get quantitative data? The answer is probably no, but what's the cutoff? So this is a plot really showing the microns between the different Z sections in that particular stack and what you want to try to achieve. So this is at 0.8 microns. So this is the lowest interval that was used for that particular Z stack. And there's over 800 objects that actually were counted. This blue bar right down here is the maximum intensity projection. So that's the collapse of you took that entire Z series and then you've collapsed it into a single 2D image. So you have to go through the process to determine, do you want to take the entire, if you're doing the 3D volumetric analysis, do you need to go down to the lowest interval, which is going to take a long time. So there are some reasons why you don't want to do that. Now, could you go to 4 micron between sections or 8? Maybe there. So you're starting to lose the cell count. Statistically, that's what matters. Is this reproducible, and is it predictive or to the biological outcome that you're anticipating? You could miss something, right? So but there's a trade-off that people I think are doing in the field, just because the amount of time it does take to do the processing and the amount of data space that it takes. So how do you optimize the image and data acquisition? I'm going to mention this. So this is something that has been introduced by in the field for a lot of companies, and I think most people that have high content imaging systems have access to something called Smart Intelligent Image Acquisition. What this is basically you're able to do a pre-scan. So you first use a low magnification objective lens to scan a large area to identify areas of interest, maybe populations based on some threshold. Maybe it's just based on morphology, maybe it's based on a protein expression or something else. And then you rescan. So this is kind of the secondary thing. You use a higher magnification lens to scan only those targeted or the cell populations of interest. And why are you doing this? The benefit of using this type of methodology for 3D imaging is you're reducing the image acquisition time overall, because you're going to reduce those empty fields as I showed you earlier. You're going to reduce the image analysis time because you don't have to process empty wells or empty fields of view. And you're going to reduce the storage volume. And you're going to increase the identifiable objects or populations for more statistical power. So here's an example of microtissue that has been plated, and people have done this, probably have seen something very similar to this. When you plate these, they don't always end up in the center. You may get lucky, you may get something like this well right here, E4, which shows the microtissue very close to the center of the well, or even D3. But many of them actually are migrating to the edge of the well. You know, sometimes that could be due to temperature deviations, and I think everyone's aware of that. When you put these in the incubator, that may happen. What the rescan, the smart intelligent design does, it recenters these objects to the center of the well. So then they're more robustly identified and being able to measure for the image analysis. The other thing that is possible is that we can get rid of wells we're not interested for saving the data or going through a whole large Z stack, because in the pre-scan perhaps we just did one 2D image, but in the rescan we took a total a Z stack of maybe several hundred of Z series. So the empty wells we don't have to worry about. There's two more that are missing, so these are probably due to artifacts that were in the well, so we can get rid of those. And this all overall is going to save time and data space. Here's another example of looking at objects that are actually in Matrigel. And so again, we can go in here and just identify those objects of interest. Here, this one's actually highlighted, shown here, but based on some threshold, size, shape, intensity measurement, we're only going to identify these objects of interest in X, Y, and Z. And here's another example where we used organ-on-a-chip model. In this case, it was from Emulate, shown here. We can see the center channel being identified here, and we did this in a pre-scan. And then there's about 250 micron difference between these two channels in three-dimensional space, so you can then go back and rescan each of those channels individually. So then you can generate this image. Now, this image is actually generated, as you can see, it's 1,200 images over 600 micron spacing, and it's from a single field of view within this particular channel. And then I think another area of interest for a lot of people now are using their high content imaging systems to actually capture slides. And I would say any high content imaging system that's out there is going to be the best fluorescent slide scanner available. And this is an example of taking a tonsil tissue. This is 353 fields at 20x, so it's quite a large image. But before this was actually even captured, it was done using the smart intelligent acquisition. So they pre-scanned it, identify where it was, and then just captured the fields within that tissue. The nice thing about it is then you could go back and then just go and identify areas of interest. So maybe you're just interested in the epithelial region, or look at the cytokeratin regions and get rid of things that you may not be interested in. You can then go into look at subpopulation analysis, for example. In this case, looking at B cells with CD20, or maybe CD4, looking at T cells, and also CD8 was done here. So this is a way to start to look at inflammatory cells that may be present. And people are using this too as far as tumors, because if you're looking at things like infiltrated T cells into tumor cells, this is a process that people may take. The other thing is it does reduce storage, and this is just an example, maybe a bit exaggerated, I think, but I think it points that comes across. If we're using Matrigel, for example, we may take 1,000 planes that could go over 500 microns because we want to get that type of resolution. If we took 225 fields of view in a 384 well plate, for example, that's going to generate about 384 terabytes of data from that plate. If we move and just use this smart intelligent acquisition, we can actually reduce that to about 160 planes. And why we reduce it to only 80 microns with 160 planes? Reason being is that that's the size of the microtissue or the spheroids or organoids that we're interested in. They're only meet that size, but they're in different Z planes. We can actually identify where they are. So therefore, we can reduce the overall storage space and time to actually acquire those images by 54 times less data and 36 less time to actually acquire those images. So on 3D visualization, there's different, one thing, these are pretty to look at. I mean, that's great. And that's nice, but you have different ways of looking at this. You can do a 3D view where you can rotate and shift and move these around. This is an example of MDCK cells to form a cyst or a lumen inside there. And that's why you see the hollow space when you do this XY view. Reason for doing this is not, this is kind of nice and you can show this off, but it's really to make sure that the image quality that you captured is what you want to see. Now you can do, there's different ways of doing this. This is a fast way really to do this, to see what it looks like. But more importantly, I think it's important to look at what the image analysis looks like. So this is an example shown here of where these cells here have been identified and the individual nuclei are actually identified here. So then you can rotate that and to actually see the, I apologize. Let me try that again. Yeah, so you can rotate that there so you can actually see where these are actually, each one of these are actually masked and identify and segmented these objects. So how do you get more out of 3D cell model system? I tell people, look, you spend a lot of time creating these 3D cell model systems, and then you take a three-dimensional stack and you make a two-dimensional image out of it for the analysis. And I understand why. It's a lot easier to do. But there's probably a lot of information in that 3D cell model. And we know that these 3D cell models are heterogeneous. We just, that's just inherent as part of it. The maximum intensity images, maybe it could be enough, and it could address the answers that you have. But when you're not sure, you could try to use these 3D segmentation tools. You can also consider the subpopulation analysis. Showed a couple examples of that. You may have to get the appropriate bioprobes to actually identify that. But I think that could be valuable for getting more out of your 3D cell model systems. I didn't go into the live cell image and limitations of the live cell imaging. Limitations, but there are some. You have to be careful that you don't cause phototoxicity of those particular samples. Light penetration is also going to be limited. Cells are not fixed, so there's going to be more light scatter because you're not removing the proteins or lipids that some of these clearing agents do. And so just got to be aware of that. And then definitely consult with the leaders in the field and consult with the companies who make these high content imaging systems. I'm sure they're going to be happy to help you, drive this for you. So I want to spend the last part really of the talk presentation, talk about cell painting, which is again a lot of traction in the field, especially in pharma, to look at phenotypic screening. And so this is actually a picture from a white paper that I actually wrote for PerkinElmer, and I put the link down here. You can look at it. It's and I don't go into it's really goes into a lot of detail around the history, what cell painting is, and some of the tools that people are using in the field, including some of the open source platforms. But in this image, I'm actually showing some of the products we use at PerkinElmer only as an illustration. But this could be any system that people are using. So in the cell painting, you're going to go through an experimental design. You're going to typically use 384 well plates, cell models, and I'll talk a little bit about which ones people are using. You're going to treat with a pertogen. Those either being chemical compounds or maybe some genetic pertogen. Antibodies are really not being used, although they could. Most people are using fluorescent dyes. I'll talk about that in just a moment. There's being multiplex, run through a high content imaging system. You go through some image analysis, machine learning. You're going to QC those images, as I mentioned before. We don't want out of focus images, for example, to be in the data set. We'll do feature extraction, and then we're going to go through some principal component analysis. Or we're also using self-organizing maps, TSNEs, and some other ways to look at the distribution of the data. and I'm going to show you that in a moment. So just some quick facts about cell painting, if you're not familiar with it. We're phenotypically profiling cells from these chemical or genetic perturbations. This comprehensive image analysis, we're generating thousands of well-level features. There's two papers to start. One in 2013, with really without naming the term cell painting. It was really coined in 2016 by Mark Bray and Ann Carpenter's paper. There's no uniform standard, and I put this with an asterisk because they're really, I think most people in the field are following the 2016 Nature Protocol paper, where they're using U2OS cells, but there's other groups that are moved away from U2OS cells and using other cell lines. The bioprobe marker selection may vary from lab to lab depending where you are, but there is a standard set of probes that were used in that paper that most pharma companies are following currently. Then there's definitely differences in the instrumentation to measure these probes, and there's different processing image analysis algorithms. This is why I'm talking. This is why I'm saying there's no uniform standard at the moment. And then there's data analytic approaches that varies. The important thing is this: we want to make sure that these results are going to be the same. So the Broad is undergoing an exercise now to address that, and we have actually participated in that study. I'll show you a little bit of that data. So this is right from the paper, from the Nature Protocol paper, that shows the dyes that are actually being used and then the target organelle. So this is the Herx nuclear dye, concanavalin A to label ER, looking at cyto14 for the RNA, looking at Floydin to look at F-actin, cytoskeleton, and then also mitogen, wheat germ, and gluten to look at the Golgi and plasma membrane, and then MitoTracker Deep Red to look at mitochondrial function. And here are some what the images actually look like of these 6 different dyes. There's a very close overlap, and I don't show that here. I do talk a little bit about the excitation emission overlaps in the white paper. But these two dyes, the concanavalin Lexa-48 and cyto14, have very close excitation emission wavelengths with approximately 85% overlap. But the important thing here is that the cells are staining 2 different compartments. So here you're looking at RNA, right? And so here you're looking more on the cytosol, looking at the ER. It's a little bit different, and that's why we're able to combine these 2. And then you're also looking at the wheat germ and gluten here. Here's the Floydin cytoskeleton, and then the MitoTracker Deep Red for mitochondria. So then you go through the features. So this is where most of the data is being generated. This particular assay is generating, and this particular algorithm is generating over 3,000 features per well. You're like, well, how do you generate that many? Well, it's pretty simple, because when you start to break it down and look at the nucleus, perinuclear region, cytoplasm, the membrane, the cells, and you start looking at the size, shape, intensity, texture, measurements, and you're not just measuring a single texture measurement, maybe measuring many texture measurement, maybe measuring many different size measurements, many different shape and measurements. And so this is how this amplifies. And some people have gone through and they have actually reduced the number of features per well to speed up the process, because this is very CPU intensive and it does take a long time to process the data. And the next step is the data reduction, really. So here's just to show the reproducibility, and these are plates from the Broad that they sent us, or 4 replicate plates with the same compounds in each plate. And you can see they cluster pretty well using this principal component analysis map with all of those 3,000 different features. So PCA is just one approach. I think there's many approaches that people are taking in the field, and I'm just showing PCA because I think most people are familiar with it. But there's other processes like random forest, there's other self-organizing maps that people are using. I think there's been a lot of interest too in the TSNE method that people are starting to look at this this particular method as well. And there's advantages to each. And so the phenotypes, I think that's what it really comes down to. Can we identify these phenotypes robustly and also reproducibly? So here's an example of DMSO controls actually shown here in the center of this cluster. And that's what the cell phenotype looks like. So that's fine. All right, so then we move to A5. pretty large distance away in the PCA map. Well, A5 shows a phenotype like this. You start to get these vacuole formations that are shown in here, different staining pattern as well. Hard to tell that you actually see all 6 colors actually in this image, but there are differences. When you move to well F4, again, large distance from the control. Morphology is definitely different from the control well. So we know that's actually good. I don't know what these compounds are. It's still blinded to us. But you can clearly tell that this is different from the control. There's well G7. Again, a different morphology that we're actually showing. Nucleus, they look larger than what you're seeing in the control. There's less cells in the well. Move to well N12. The cells are more elongated. The nucleuses are more elongated formation. So definitely a different phenotype shown here. And then we move to well B9. The nucleus look more multinucleated in shape, and the cells are much larger in size. So you can see that we can generate these different phenotypes here, and these are going to cluster likely to these compounds having the same mode or mechanisms of action. And that's the key behind the cell painting process. So the conclusions and really take home that I have for you really are to, and I think you probably heard this over again. I think Nate had mentioned this earlier. You want to generate robust, reproducible, and repeatable assays. Again, the images of the data, we want to generate the best images possible. We want to enrich the data findings with machine learning and AI. Know the limitations of the technology. Wide field can't do the same thing as confocal. Confocal cannot do the same things as the light shoot instrument if you're using that. Smart intelligent image acquisition saves time and space. Use it if you have it. Seek guidance from the AGM and peers. So please, review. We have 4 chapters. We have more coming for high content, and so please, think about using that as a good resource. So this is my last slide, and I'm going to take questions after this. But again, this is that chapter on assay development guidelines for image-based screening. I've given this talk before. I go into a lot of details on all those different approaches, things that you want to think about, like cell lines, plates, splitting, passages, that thing in a high content image assay. And I believe that information has already been pre-recorded already. So I wanted to show a little bit different data today. There's a book that Paul Johnston and I edited, and it's out that may also be useful for you. I do want to mention SBI2, if you're not familiar with the organization. Right now they have an ongoing free educational series. They started after their annual meeting in September. So they actually have a talk that's ongoing right now. So I didn't want to tell you about that, so you could listen to me. But they also have talks on December 9th and January 14th that you may be interested in. And again, this is my contact information. So I'm going to stop there and take any questions.
Thank you very much, Joe, for this wonderful presentation. I do not see any questions in the chat box currently, and we're 5 minutes over the time anyway. So what I'd suggest, if you could stay few minutes and keep an eye on the chat box and the Q&A box, and answer the questions. So please go ahead and ask all your questions to Joe. He's going to be here for a few minutes to answer your questions. So we have a break now. So this concludes our morning session. And we're going to have a break for lunch. Anyone that is was not able to secure a spot at the roundtables, we will reconvene at 1:30, and whoever was able to secure spots at the roundtables will be running from 12:30 till 1:30, and you should have received emails by now guiding you how to access the individual roundtables. So please go ahead and log in to those links, and then just log out and get back into this main link when the roundtables are over. So again, we will reconvene at this meeting link at 1:30. Abigail, is there anything else you would like to add?
No, thank you so much, Serene. If anyone has any issues getting into the roundtables, I will be available by email and also via the chat here in this main room. Thank you very much and enjoy your lunch.